Hello everyone, my name is Elise Richardson and I'm a new PhD student at NC State. I'll be presenting my research from my master's at the University of Florida in the Keiser lab. I'll be presenting pathogen prevalence and individual variation in tick host-seeking behavior. Vector-borne diseases account for a large proportion of infectious diseases that affect humans and wildlife globally, and with climate change expected to create conditions that expand a vector's geographic range and habitat, it's become extremely important to understand the disease dynamics of vector-borne diseases. Many studies have shown changes in behavior of vectors when they become infected with a pathogen. Examples of this have been seen in mosquitoes, sandflies, fleas, and kissing bugs. The same phenomenon of changes in behavior associated with infection have been seen to occur in ticks. Ticks infected with Lyme disease have been shown to spend longer amounts of time questing, and a study that infected ticks with tick-borne encephalitis virus showed that when infected, ticks engage in increased questing activity and motor activity, aggression, and are willing to withstand obstacles to obtain a blood meal. The role of consistent individual differences in behavior in host-parasite dynamics are usually looked at from the perspective of the host behavior. However, there have been some recent studies that have focused on individual variation in host-seeking behaviors in ectoparasites. These studies have been done on kissing bugs, Ixodes ticks, and an ectoparasitic mite. Understanding these individual level variation in disease vector behavior, especially in medically important vectors, could give us a better understanding of disease dynamics, transmissibility, and the impacts that they could have on public health. My research questions are as follows. How does questing behavior vary across populations? Do ticks display individual variation in questing behaviors? And then, does pathogen prevalence affect this behavior? The tick that I'm conducting my studies on is the Lone Star tick, also known as Amblyomma americanum. It's considered to be one of the most aggressive ticks, and as you can see over here on the top right, this tick is a female, and you can tell by the white star or dot on her back. Lone Star ticks are known for transmitting human monocytic ehrlichiosis, tularemia, heartland virus, and many Rickettsia spotted fever illnesses. We collect our ticks using a tick drag, which is a white canvas sheet that we drag through the habitats. This is a useful way to collect ticks from a large area, and it specifically collects questing ticks. Throughout the summer, I collected a total of 902 ticks. The host-seeking behavior that I'm studying is called questing behaviors. In order to quest, ticks will climb up and hold on to leaves or tall grasses with their first pair of legs outstretched, waiting for a host to walk by. When this host does brush up, the tick will climb on and find an appropriate spot to attach and take a blood meal. I conduct all questing assays in an outdoor controlled environment within a questing arena, which is a see-through planter container with a wooden dowel held by putty in the middle. Each tick is placed at the bottom of the rod and is given 10 minutes to quest. Questing heights and durations are recorded throughout this 10-minute session. Unfortunately, due to coronavirus lab limitations during my experiment, I was not able to conduct my questing assays in the lab as was originally planned, so instead I used my porch. It was a location that was still outdoors but covered and protected from rain or severe wind. You can see this setup in the upper left picture. After questing assays were complete, we extracted DNA from all ticks individually, then completed conventional PCR to screen all ticks for both Rickettsia and Ehrlichia species, but we found so few Ehrlichia infections that they were not included in the analysis. However, we did find many Rickettsia amblyomatis infections. Rickettsia amblyomatis is a part of the spotted fever Rickettsia group and was previously known as Rickettsia amblyomi. Although this is considered a more recently discovered pathogen, so far current publications on the species of Rickettsia have alluded to it being a possible human pathogen. There are also other publications that have found that Rickettsia amblyomatis could be inhibiting or complicating a Rocky Mountain spotted fever diagnosis. Collecting ticks from similar habitat types allowed us to analyze the data at a population level and at an individual level. We collected ticks at parks and hiking trails here in Gainesville, Florida between April and August of 2020. Here is a map of the 5 sites that we collected at. 4 of them are parks, and 2 of the parks we picked were counted as one population due to the close proximity. We also included the Ordway-Swisher Biological Station in the analysis as an additional population, which we used in a previous study. I collected at each site twice a month for a total of 5 collections per site throughout the summer. And here are some pictures below of the 4 different parks so you can see how similar the habitat types are. All ticks went through the same questing assays, but a subset of them, 253 of them, were tested for 3 days in a row to test for repeatability in the questing behavior of the ticks. Our findings show that there's actually quite a bit of variation with each individual tick. As you can see from this table, 27% of the ticks used in this experiment did not quest at all, 30% quested for 1 of the 3 days, 29% quested for 2 of the 3 days, and 14% quested for all 3 of the testing days. This figure shows the repeatability estimates for 3 questing traits: questing likelihood, questing height, questing duration, which were all measured 3 times in a repeated questing assay. The 2 values in the gray bar represent the repeatability of questing likelihood estimated for uninfected and infected ticks separately, and points represent repeatability. And intervals that overlap 0 represent non-repeatable behaviors. So this shows that all traits except for the total time spent questing were found to be repeatable behaviors. Specifically, we found questing height to be repeatable, and this could have an effect on host preference. Although we did not find differences among populations with the questing height, this could be because a lot of the hosts in Florida are just similar, and it's possible that at a larger scale, we could have seen differences. The top of this figure shows the infection prevalence between the nymphs and adults. The bottom of the figure shows the proportion of ticks questing of adult and nymph ticks. Each point represents the average of each site population. At a population level, we did not find any differences between the locations with infection prevalence, nor did we find among population differences in questing behavior. Although we did find that adult ticks are more likely to be infected with Rickettsia amblyommatis than the nymph ticks, and we found that nymphs are more likely to engage in questing during our assays than the adult ticks. This figure shows the total time spent questing for adult and nymph ticks with the infection status of Rickettsia amblyommatis shown as well. Blue bars represent the uninfected ticks, and orange bars represent the infected ticks. The number in each bar is the population size. From this figure, it can be seen that infected adults spent more time questing than the uninfected adults, but this was not considered a significant difference. The infected nymphs spent significantly less time questing than the uninfected nymphs. Arthropod vectors represent a key point in the life cycle of the parasites that they transmit, and understanding their behavior and physiology can expand our ability to understand and potentially prevent disease outbreaks. This is, to our knowledge, the first study to look at repeatability and individual variation in tick host-seeking behaviors, and we have found that ticks infected with Rickettsia amblyommatis spend less time questing than uninfected ticks, and that these trends were dependent on the life stage of the tick. I'd like to say thanks to my advisor, Nick Keiser, and the whole Keiser lab for all of their help and encouragement. Thanks to Dr. Estelle Martin and Caitlin Taylor for their molecular assistance, and thanks to David Buttrick for assistance in collecting ticks. The Lone Star Ticks and I would like to say thanks, and we appreciate your time. Thanks so much for listening.